I would like to introduce Dr. Hoffman, who's the founder of Origami Therapeutics. Dr. Hoffman brings more than 25 years of expertise in CNS drug discovery. He developed more than 30 assets, advancing into clinical trials across various domains, including cystic fibrosis, metabolic disease, neurology, pain, and psychiatry. As a scientific advisor for the Tau Consortium, he focuses on alleviating diseases associated with Tau pathology like Alzheimer's disease. The talk today will be dementia in women.
Can you hear me?
Okay,
Then I
take
can
it away,
hear the
Dr.
echo.
Hoffman.
Thank you so much for the very kind introduction. So today I'm going to talk to you about a bit about a topic I don't usually talk about, so I don't usually give too many very broad presentations around dementia. So I'll look for the feedback. So the title of the talk is Neurodegenerative Disease, the Quest to Make It a Thing of the Past. The subtitle, Women and Dementia. And so hopefully that will be interesting. So here, before I start my seminar, I want to take a little bit of a pre-seminar poll. How many of you know someone with dementia, any kind of dementia? Look around. I'm sorry that I'm not sure whether the people online can see, but if you keep your hands raised, there's an awful lot of people. Now, maybe we have a selected group, so it's a selected group. Okay, your hands are up. Tell me, how many of you, your someone actually is, might help them. Yes. Okay, so I'll just keep going. So how many of you who had your hands raised, your someone is female? So I don't think any of you are going to be surprised by some of the statistics I share with you today. So, as with many diseases, we have an increasing burden of dementia worldwide, and this is just representing numbers worldwide, different years. So 2030, and you can see the huge increase as we move to 2050. This is not consistent. So just in general, here's how dementia is defined. A catch-all category of diseases affecting memory and brain processing, including Alzheimer's disease. So as you might imagine, and for those of you who have loved ones or friends with dementia, devastating consequences, economic, societal, and cultural. So here's some more frightening statistics. Numbers are rising quickly. By 2050, 1 in 5 will be afflicted. Another 1 in 5 will be a caregiver or will be paying someone else to be a caregiver. So you can see how both the economic, societal, and cultural factors feed in. So does dementia differentially affect women and men? Well, I suppose I wouldn't have asked the question if the answer weren't yes. So men tend to be diagnosed with vascular dementia and Lewy body dementia more often. Women, on the other hand, are more often diagnosed with Alzheimer's disease. And my guess is that that was reflected in all the hands we saw in the pre-seminar poll. So, more concerning statistics. According to WHO, neurologic disorders are the second leading cause of death after heart disease, accounting for 16.5% of global death. Furthermore, in the US, 2/3 of those living with dementia are women. In Australia, nearly 2/3 of all dementia-related deaths are women. US women over 60 are 2 times as likely to develop Alzheimer's disease as breast cancer. And I wasn't able to see all of the numbers from our previous speaker to compare whether my numbers and her numbers match. In England, Wales, and Australia, dementia is the leading cause of death for women, displacing heart disease from the top spot. Think about that. So this is a really big problem, not just for women, but this is a really serious issue. So Why is AD more prevalent in women? Here I have a quote from Dr. Aziz. For a long time, it was thought that rates of dementia were higher in women because women have tended to live longer than men. Actually, Elena and I were just we were talking about that at a different talk. Well, maybe there's more, there's a skew towards more cardiovascular disease at 85 plus because there are more women. I don't know, but upon reanalysis of the data, researchers have felt that it's more just that there are other differences accounting for the increased risk. So, several possible risk factors. Hormones. We heard about differences in hormones. Estrogen may be protective. Estrogen levels drop during menopause. We heard about that in the previous talk. Early menopause may increase AD risk. Tau. Tau is one of those proteins that you hear about. You hear about A beta plaques and tau tangles. Tau is a protein that accumulates and may cause Alzheimer's disease. And actually, tau somehow acts differently in women's brains. Don't have nice graphs to show you because these are just correlations at this point, post hoc. And so what different means is still yet unclear. And I think we'll get to the point that there's a lot we don't know. Genes. So ApoE4 is one of the major risk factors for AD. However, women have an ApoE4 gene, they are more likely to develop AD than men with ApoE4. So, sex differences in dementia are only just being recognized. That's why I don't have a lot of these wonderful, longitudinal graphs. Love to have it, but I think we have a long way to go in this area. So, but the consequences could change the way we fight the disease, and that's why it's so important for us to get a better handle on what's really going on. And this holds for men and women alike. What about vascular dementia and Lewy body dementia? So, here are some of the risk factors. Vascular dementia, blood flow disruption, heart disease, diabetes, high cholesterol levels, smoking. We've heard this list over and over this morning. Lewy body dementia, general diagnosis, ages 70 to 79, 4 times more men. Frontotemporal dementia, now this is generally much earlier onset, age 40, 65. I won't embarrass anybody by asking how many of you are over 40. I am. So that's pretty young in my book, but affects men and women equally. So, and as I think we've heard, this long list under vascular dementia gives rise to risk for many diseases, but also are risk factors in Alzheimer's disease. So, other risk factors that affect women more than men: surgical menopause, pregnancy complications such as preeclampsia. That's been linked to cognitive decline. Depression has been linked to onset of Alzheimer's. Sensory deficits. I think we hear more and more ads, hearing aids and such. But deficits in vision and hearing are considered precursors to a risk for Alzheimer's disease. We've heard about problems with sleep disturbances. Same is true here. Sleep disturbances are often linked to depression and other mental illnesses as well. Social isolation, that's akin a bit to sensory deficit. And then social roles. And I want to emphasize this point because I think we don't think about caregivers. But caregiving is a risk factor for AD. 60 to 70% of all unpaid caregivers looking after someone with dementia are women. And so this is an even greater risk. But I just want to stop here and say, we often don't think about the caregiver and the toll that being a caregiver takes on an individual. So I'll just make a pitch and say, yes, we need to understand these better. But we also need to take care of our caregivers. So New sex-stratified research will help develop more effective treatments and, most importantly, preventive practices for patients. One thing that I like to talk about is the fact that it's better that you prevent loss of brain cells than have to try to replace them. So I think we have to think very carefully about how is it we diagnose and get in early. Again, theme that we heard multiple times this morning. So, the quest to make neurodegeneration a thing of the past. So, there are three main categories that I've had a number of discussions about, even just in January. And I want to just cover those so that you can think about what is it that we need to think about. Where are the gaps? And there are lots, and the gaps are big. So, defining disease. So we need to be looking at biology rather than just symptoms, because I think everybody understands that, you can have 20 people in a room with a headache. The headache came from multiple different reasons. And I'm taking very simple. Yeah. Genetics. There's a lot of discussion about genetics, about genetic linkage, about genetic modifiers. I think we also need to be careful in defining what is the cause of the disease and what is associated with the disease. So if we hope to treat forms of dementia, we need to understand what the cause is so we can fix it. Defining disease stages based on natural history. So for those of you who are very familiar with oncology, you know that there are generally 4 stages of disease. We've heard a little bit about that from on the breast cancer topic. But until recently, we haven't had those sorts of very defined disease stages with clear milestone hallmark measurements, things that you can quantitate. Because if you can't measure it, then there's no way to know about it. How are you going to fix it? And again, I'm going to mention early diagnosis. Second big bucket is biomarkers and digital measures. So I don't have my phone with me, but all of you have your phone. Did have it with you before. But that little box, used to occupy a space this big when I was taking computer classes at NYU a few decades ago. And so that's a really powerful tool. I mean, everybody's playing games and communicating. There are a lot of things that we can measure remotely on phones or iPads or something like that, or your Apple Watch. What we're missing is more frequent measurements. So I don't know how many of you are familiar with clinical trials and how things go there. But, usually those enrolled in the clinical trial come to the clinic, at set times, maybe once a quarter, maybe more than that. But the fact is that that's not a very frequent way to measure. And when people come to the clinic, they want to do their best. And so it's pretty clear that you don't get a really good sense of how well the person does. I think anybody who's even had the flu, one day is good, and then the next day, like crap, right? So neurological diseases are like that as well. And so we need more frequent measures. We need additional measures. We need to look at multiple measures, multiple outcomes. So the other piece is that when you hear biomarkers, most people are talking about validated biomarkers, things that you're going to use as surrogates for the disease itself. And those are great. It takes a lot of effort to validate a clinical biomarker, but there's so many experimental and non-validated biomarkers that are legit and that we can use for decision-making purposes. So I'll just say my observations are that there are a lot of phase 2 trials or phase 1, phase 2 combined trials where things have not been done as rigorously as they ought to be. You don't know what dose to give, not quite sure about exactly what to measure. And gosh, you've come this far. And so we've just got to push forward into phase 3. And then you have the big loud failures and the patients are devastated. And so these are sorts of things that we really just have to incorporate and be very disciplined about. Finally, there's a lot of talk about combination therapies, drugs with digital therapeutics, video games, as therapy, things of that nature. But again, I think we have to carefully think about preventive behaviors. So what can we do? Diet, exercise, we've heard about not smoking, and a variety, sleeping well, those sorts of things. Because in the end, preventing neurodegeneration has a better likelihood of outcome. So these trends are being actively pursued, so I don't mean to say that this is new and nobody knows about it. I think lots and lots of people know about it. People are working. But compared to other diseases such as cancer and heart disease, dementia has historically been underfunded. The other piece, and some of you may know this from the recently approved MB and the Aduhelm that was previously approved and now, as of today, withdrawn from the market by Biogen. Everything's been focused on A beta. And what's clear is there might be something there. We need to do better. We need more diverse approaches, and that needs to be further encouraged through investment. So let me come back to one thing, and that's leveraging genetics. So I mentioned causative versus associative. But the one thing we do know about the value of genetics is that if you leverage genetics in your drug target, you have an improved likelihood of success. And in fact, that's been estimated to be 2 to 8-fold higher probability of success. So I'm going to move on and talk a little bit about an example of how do we put a lot of the information that I've talked about so far together. So how do we approach developing curative medicines for neurodegenerative diseases? So this I've taken from a couple of references, and this is the proposal for the pathway to best in class. I talked about genetics. The other piece here is that when you go pick your target, you think you know, but you don't really know whether that's the right target. So, you could have 1,000 genes involved in 1,000 different proteins involved in whatever disease. One way to not sort of just gamble and take a pick is to look at phenotypic screening. And I'm a big proponent of that. I'm going to show you an example of why I am. But the important piece here is that you are measuring something that you want to do in human disease. So the most important thing here is you can make drug candidates that look safe and you can put them in the clinic all you want. If you can't measure them in clinical trials, then it won't help you. It doesn't translate to human disease. Likewise, everybody's wasted their time and energy, and now you're asking the patients to put themselves at risk. So I don't know, but I like to be successful. When I spend my time doing something, I want something to work. And so I think we need to go back and reconsider phenotypic screening. That's a path to get both first in class and best in class. And with the new advances in proteomics and genetics, gene editing, we're able to, once we found molecules that do what we want them to do. So I'm going to tell you a little bit about what I'm doing at Origami Therapeutics. So Origami was founded on an innovative small molecule approach, functionally for neurodegeneration. We have unique insights. A lot of this comes from my experience working at Vertex on the cystic fibrosis drugs. And the key here is you screen using clinically relevant endpoints. That means you're measuring an effect the drug should have in patients. And you use human disease models. You use human cells. This is critically important. It had to be critically important in cystic fibrosis and in the positive phase 3 trials for a non-opioid pain medication that was just announced yesterday. also from Vertex, something that I've worked on. So we use patient-derived human disease models, and the key here is prioritizing what you've screened out. These are small molecules. The key here is get into in vivo PK/PD studies as soon as possible to figure out which of the molecules you should be bothering to go after. And this is how you get very quickly to demonstrate in vivo proof of concept, in vivo efficacy, and now you just have to make the molecule better. So this is a quick way to de-risk a small molecule effort. So our first indication is Huntington's disease. So there are no effective therapies for Huntington's disease. It's a devastating neurological disease. It's an autosomal dominant disease, so you only need to inherit one copy of a parent in order that you will eventually develop Huntington's disease. The average age of onset is pretty young, 35 years old. I mean, you're at the prime of life, your career's going, having your kids, and this is really devastating. So, but the point of going after Huntington's was twofold, and I've already mentioned these. It's a genetically driven problem. The mutant protein is the direct cause of the disease. In addition, because it's a genetic disease, we know who to treat. And I'll get on to that as well. So as I said, you got to know what you got to fix, and you've got to be able to recruit and run the trials in the right patient population. So other keys to Huntington's disease is that this is a huge protein. It's expressed in every single cell of your body, from conception to death. The wild type protein is critical. And so you can't go in there and blow up the entire protein, mutant and normal. That's not going to be consistent with life. So suppressing or removing the mutant protein restores cellular function in animal models, and we and many others believe that elimination of the mutant protein will drive clinical benefit. I don't know if I'm going to get this. People can't see it on the screen anyway. But what you see in the lower right is an HD brain post-mortem versus a normal brain. And you can see that there's a huge amount of loss of brain tissue. What I'll also tell you is that this is a genetic disease. It's a systemic disease. So your muscles are affected, your immune system, your gut, your liver, virtually every organ. And so we need a solution that will treat the entire disease, and that's small molecule. So, challenge, how to remove the mutant Huntington protein. I kind of gave away the bottom line here. So most of the common approaches that have been used already are antisense oligonucleotides, gene therapy in the form of primarily AAV vectors, and protein therapeutics such as antibodies. And here are the issues. I told you it's an autosomal dominant mutant protein, forms toxic fragments. Delivery to the site of action, you have to be able to get into all or most cells in order to stop progression of the disease. In addition, most of what's been done so far is reduced both mutant and wild type protein with the idea that that's okay. It's only important in development, in fetus development, and so it's okay if we knock the snot out of it. Probably turns out that that doesn't look to be true. So these two issues have really contributed to the shortcomings in HD clinical trials. So what has origami done? We have small molecule direct degraders that address the limitations of other approaches to functionally cure Huntington's disease. Selective for the mutant, spare the wild type, should allow us to restore cellular function. They're brain penetrant, orally available. They have favorable drug-like properties, clean preliminary toxicological profile, and the molecules are simple and straightforward to manufacture. So I told you I'd talk a little bit about the clinical and regulatory path. It's reasonably straightforward. So HD is an orphan disease. As such, it's subject to expedited regulatory paths such as orphan, breakthrough, and fast track designations with active patient advocacy groups. And I think most importantly, in our phase 1, we'll have a single ascending dose, multiple ascending dose in healthy volunteers, but the third arm will be proof of mechanism and biomarkers as part of the multiple ascending dose in HD patients. So at the end of phase 1, we're going to know whether the molecule at least works mechanistically. So what's been done quite a bit with some of these biologics is phase 1 and phase 2 are combined. Unfortunately, that means that there's limited dose exploration, and so go into a somewhat limited phase 2 trial. Here, we would be prepared now to go into a real live full phase 2, figure out whether the drug is going to work. So I'm going to stop there. Happy to answer any questions about dementia in general or our approach. Thanks so much for your attention.
Thank you so much, Dr. Hoffman. We'll open it up to questions from the audience. Questions? We have time for questions or so? In the back.
What kind of degraders that you are talking about here? Is it PROTACs or molecular glues or LITACs or APTACs?
So see, these are not proximity induced. So PROTACs, as well as all the other ACs, are proximity induced. And as such, they hijack natural degradation pathways. Our molecules are direct degraders. They're small, and they basically kick open the structure of the protein so that it can more easily be degraded. So I'll just comment that for cancer, hijack a couple of pathways is fine if you compromise the health of the cell, because in the end, you want the cancer cell to die. That's in chronic disease, including neurodegeneration, that's exactly what you don't want. You don't want cells dying. You want to return cells to normal homeostasis as close to as best you can. And so that's why we think that our direct degraders have a decided advantage over PROTAC.
Thank you.
Any more questions? Oh, yes.
Thank you, Beth. I like your statistics at the beginning as well. I hope you share them with us. One question, is your technology also then applicable later on for other neurodegenerative diseases? I think you insinuated that, but the same principle, probably a different molecule then, so it's different genetics or different degraders for other, but the same principle. Basically, you have a platform, right?
Yeah, that's how we see it. It's a biological platform, and with quite a bit of understanding about the biology, the deep biology, using human systems. But yes, we went with Huntington's as our beachhead, basically to show that our approach worked, and we're hoping to jump into other neurodegenerative indications as well. Thanks.
You mentioned that dementia has been traditionally underfunded, and I think part of the reason is perhaps because of a lack of approved targeted therapies. And you mentioned that Leqembi and all these other medications have been recently approved for Alzheimer's disease. So I'm wondering, what could manufacturers of those companies or those medications, or like even the wider community, can do to bring awareness to this issue? Thank you.
The issue meaning more funding?
More funding, and also more awareness in terms of the gender bias or predisposition in dementia.
Yeah, so I think, as with many things, success brings more people in. And so, I think a lot of us are hoping that the approval of Leqembi, where you actually do see a lessening, will bring more funding. into neurodegeneration. Times have not been good in the past 18 months for fundraising for everybody across the board. I think, I think we hope this, and we'll see. So maybe there's some people in the audience who can help us fix that.
Okay, thank
Thanks
you
for the
so much,
question.
Dr. Hoffman. Let's give her another round of applause.